Good afternoon, everyone. I'm Shreya, undergraduate student from Coorg Institute of Dental Sciences. I'm here to present my paper on potential health benefits from the husk of cocoa berry. Cocoa berry, otherwise called as Theobroma cocoa, is largely used in chocolate industries. As chocolates have always been a fan favorite among both young and old. But it is the dentist who is raising the concern about chocolates. We know that sucrose is proved to be the arch criminal in the etiology of dental caries. Chocolates are the combination of both cocoa and sucrose, in which they contain greater amount of sucrose along with cocoa. So our focus went on cocoa berry. About 70% of the world's cocoa bean come from 4 West African countries. These are Ivory Coast, Ghana, Nigeria, and Cameroon. The Ivory Coast and Ghana contribute to the 50% of the world's cocoa production. So cocoa beans are widely used in making chocolates. It is dried, fermented, and roasted to yield cocoa chocolate and cocoa butter. So the aim of our experiment was to assess the available biochemicals and antioxidants in the husk of Theobroma cocoa. The outer part was called as cocoa pod or husk. The inner part is called as cocoa bean. Coming to methodology, collection of cocoa berry from cocoa plant. The outer husk was separated from the berry. The husk was sun-dried for 3 days and grounded into powder. 5 grams of grounded powder was mixed with 100 ml of distilled water. Then it was boiled for 15 minutes, following which the filtrate was extracted. Then this cocoa husk extract was used to perform 2 tests. First one, test for phytochemicals, and second one, test for antioxidants. First one, test for phytochemicals. Phytochemicals like alkaloids using xenontian reagent, phenolic compounds using ferric chloride, flavonoids using alkaline reagent, carbohydrates using Benedict's reagent, proteins using biuret test, and terpenoids using Salwonki's test were tested. Second one, test for antioxidants. Antioxidant property of cocoa husk was evaluated by 2 assays. First one, total antioxidant activity. Second one, ferrous reducing power assay. Coming to results, phytochemicals like alkaloids, carbohydrates, and terpenoids were found in the husk of Theobroma cocoa. In total antioxidant assay, 100 grams of husk demonstrated antioxidant activity equivalent to 2,400 milligrams of ascorbic acid. In ferrous reducing power assay, 100 milligrams of cocoa husk extract had free radicals reducing capacity equivalent to 3,104.5 milligrams of ascorbic acid. From the year 2014 to 2019, in different parts of the world like Brazil, Ghana, Nigeria, Indonesia, Croatia, they conducted the experiment regarding cocoa pod husk. 2014 in Brazil, fermented cocoa pod husk extract was used to perform the experiment. Their aim was to study antibacterial activity, and they found phenols, steroids, and terpenoids. In 2015, their aim was to study the properties for the use of nutraceutical and functional pharmaceutical. 2016, their aim was to study the application of antimicrobial, antioxidant, and larvicidal agents. 2017, their aim was to study the antibacterial activity against Enterococcus faecalis. In 2018, their aim was to test for phytochemicals. They found theobromine, caffeine, and flavonoids. In 2019, their aim was to determine the minimum inhibitory concentration of cocoa extract against Enterococcus faecalis biofilm. Again, in 2019, their aim was to determine the evidence of the differences between root canal wall cleanliness when treated with 8% of propolis extract. From the previous discussion, the summary was that cocoa pod husk was found to have high nutritive value, that is dietary fibers and proteins. It exhibited antibacterial and antimicrobial activity. It contributed to antioxidant properties, and it can be nutraceutical and functional pharmaceutical. In our present study, phytochemicals identified from cocoa pod husk were alkaloids and carbohydrates, which were not reported in earlier studies, along with terpenoids. In our present study, antioxidant properties were attributed to the presence of ascorbic acid. In 2016, in Nigeria, country in West Africa, When experiment was conducted using cocoa pod husk, the pod husk extract was found to have some antioxidant properties. The dietary intake of small doses of alkaloids have therapeutic uses as muscle relaxants, tranquilizers, painkillers, and antimicrobials in human body. Carbohydrates are the main source of body's energy. It fuels our brain, kidney, heart muscle, and central nervous system. Terpenoids were found to be useful in prevention of several diseases, including cancer, and also have antimicrobial, antiviral, anti-allergenic, antispasmodic, anti-hyperglycemic, and immunomodulatory properties. The presence of phytochemicals like alkaloids, carbohydrates, and terpenoids initiated us to explore for the presence of antioxidants. Antioxidants scavenge free radicals from the body cells and prevent the damage caused by oxidation. Antioxidants such as vitamin C and vitamin E boost our immune system. Antioxidants lower the level of low-density lipoprotein, thus preventing plaque deposition in the blood vessels. So the best food sources of vitamin C are yellow bell peppers, guava, lemon, strawberries, papaya, broccoli, kale, grapefruit, orange, and kiwi. To this list, probably we can include the cocoa pod husk in future. So cocoa pod husk suggests to aid in metabolic system of humans, that is alkaloids, probable nutrient value of husk, that is carbohydrates, role to combat diseases in the body, that is terpenoids, a potent antioxidant similar to ascorbic acid, that is vitamin C. So Theobroma cocoa or cocoa pod husk can be of health benefits. Thank you.